The unique divergence of the mammalian lineage began with the synapsids, a diverse group of amniotes distinguished by a single opening low in the skull roof behind the eye. The earliest synapsids, the pelycosaurs, evolved and diversified in the late Carboniferous and dominated their ecosystems well into the early Permian period. The term pelycosaur, however, doesn't actually refer to a single lineage. To illustrate what I mean, let's take a look at this cladogram. As you can see, the synapsids are a single lineage, separate from the sauropsids, which include living birds and reptiles. As such, Synapsida is termed a monophyletic clade. The therapsids, which I'll be talking about in this episode, are also monophyletic, as are the cynodonts, subjects of episode 3, and living mammals. Pelycosaurs are actually a collection of pre-therapsid synapsids. This grouping excludes some descendants of the nearest ancestor, and so is termed paraphyletic. Generally, taxonomists prefer to use names that refer to monophyletic clades because they reflect common ancestry and evolutionary history. But sometimes, paraphyletic terms have a history of traditional usage that makes them practical in certain contexts, such as this series of educational videos. The words reptile and fish are also commonly used terms that refer to paraphyletic groups of species. One of these pelycosaur lineages, the carnivorous sphenacodonts, include the famous sail-backed Dimetrodon, which is often popularly confused with dinosaurs. The name Dimetrodon actually means 2 measures of teeth, and refers to the creature's distinctive dagger-like canines. These contrast smaller shearing teeth in the rest of the jaw. All sphenacodonts have this feature, and were the first amniotes to have such obviously differentiated teeth. In combination with more developed jaw muscles and a progression towards larger body size in the early Permian period, this suggests that sphenacodonts evolved into highly specialized hunters. The large sail on some later members like Dimetrodon may even have been an early thermoregulatory structure that allowed the animal to raise its body temperature quickly at sunrise and take advantage of other slowly rousing synapsids. Part of being a better hunter is being physiologically more efficient with energy resources. And the environmental conditions throughout the Permian period seem to have encouraged the evolution of a truly remarkable innovation, the ability to metabolically control internal body temperature. This stage in mammalian evolutionary history is perhaps the most important. The transition from the basal synapsid condition of pelycosaurs to a mammal-like life history plan is comparable in its profoundness, I feel, to the transition of early archosaurs into birds. In the early to middle Permian period, a lineage called Therapsida diverged from within the sphenacodonts. The grade of transitional specimens between basal sphenacodonts and derived therapsids is in fact so abundant that the classification of some specimens as either therapsid or sphenacodont is highly debated. This is a level of detail more approximating our ideal full pedigree situation, and gives us a rare glimpse at incipient lineage formation, where taxonomic categories lose the power they, by definition, derive from hindsight. Therapsids did not evolve endothermic abilities directly. There's no immediate environmental pressure that can select for the nebulous feature that is endothermy as a whole. Rather, therapsids are distinguished from other synapsids by an array of key characters that are all correlated with each other. All these characters have evolved in parallel, with the result being homeostatic regulation via increased metabolism. This kind of evolutionary process is termed correlated progression. These changes seem to have been initially driven by simple adaptations for better carnivorous hunting efficiency, such as organized incisors that fit well against each other and long canines for a more effective bite. Structural adjustments also allowed improved jaw stabilization and musculature for greater bite forces. These included a reduction of the quadrate, a jaw joint bone, to expose more areas of the squamosal and brain case for the attachment of facial muscles. Being more efficient at obtaining food naturally leads to having more energy reserves, which in turn allows for increased metabolic function, and all the advantages a more energetic lifestyle provides, such as even better hunting and escape abilities. This would have, in parallel, put selective pressure on other adaptations for more efficient metabolism. One of these adaptations is the ability to eat and breathe at the same time. Being able to eat while breathing is something we take very much for granted, so you may be surprised to learn that many animals can't do this. As mammals, we have a skull structure that separates our nasal cavity from our oral cavity. That structure is the secondary palate. But most tetrapods have a nasal cavity that is continuous with the oral cavity, with no secondary palate separating them. This means that respiration is impeded during the ingestion of food. In early therapsids, we see the beginnings of a secondary palate forming through the extension of the maxillary and palatine bones. This has effectively pushed the internal opening of the nostrils back farther in the upper jaw. In therapsids, we also observe adjustments in the skeleton and skeletal musculature to allow a wider range of motion in the joints, and more energetic movement in an erect posture. This contrasts the sprawling posture and lateral movement pattern of early synapsids, amphibians, and many living sauropsids. The lateral gait is in fact a remnant of the swimming motion of fishes. The contrasting movement pattern of the mammalian erect gait is probably best exemplified by cetaceans, who swim by undulating their bodies up and down, not side to side like a fish. This change in stance may also have aided lung ventilation, encouraged the evolution of increased lung volume, and the development of diaphragm muscles, all features that enhance metabolic efficiency. Finally, we also see important changes in the bone structure of therapsids. Pelycosaurs generally have poorly vascularized bone, like many ancestral tetrapod groups, and a slow bone growth pattern. In therapsids, including mammalian descendants, we see a highly vascularized fibrolamellar pattern that indicates rapid bone growth, a clue that therapsid development was very different from their closest pelycosaur relatives. All of these adaptations together that improve energetic efficiency via higher rates of resource utilization effectively feed back on each other in an evolutionary cycle that ultimately results in greater capacity for an endothermic lifestyle, where metabolism is used to regulate internal temperature. The advantages provided by an endothermic lifestyle are many and include the ability to capitalize on wider geographic and temporal ranges, complex internal physiology, greater aerobic capacity, and increased juvenile provisioning and growth rate. That we observe all of these important changes incrementally in each evolutionary grade represented by fossils from the pelycosaurs through late therapsids reflect the organism operating as an integrated whole, far removed from the mechanical concept often associated with evolution. What's more, the evolution of other characters that are unobservable in most fossils can also be safely implied. A skeleton, skull, and bones like these mean that this animal had a higher metabolism, and so could use that metabolism to help regulate its internal body temperature. Therefore, even though we can't see them, this animal also very likely had a double circulatory system with a 4-chambered heart, precise chemoregulation, a larger brain, and cared for its young in some capacity. In this most important of transitional progressions, we are also fortunate in having robust data for reconstructing paleobiogeography. The fossil record shows the first appearance of therapsids as a geologically rapid radiation in the Middle Permian period, about 267 million years ago. However, given the magnitude of evolutionary changes in the lineages leading to therapsids, it's unlikely that they both originated and diversified so extensively in only a few million years, an evolutionary model known as a short fuse. A long fuse model is more likely. Under this model, it's been hypothesized that a sparse therapsid lineage arose in the early Permian about 300 million years ago that was then followed by a radiation in the middle Permian. The low initial diversity would make fossil deposits from that time rare. This predicts eventual discoveries of earlier therapsid fossils, which may even include some currently debated finds, and an environmental trigger of some kind for a mid-Permian radiation. This long fuse model is in fact very congruent with what we know from paleogeography. The early Permian was marked by globally increasing temperature and seasonal patterns as Pangaea shifted northward and glaciations on the southern supercontinent of Gondwana retreated. Based on discovered fossil locations, late Carboniferous and early Permian pelycosaurs appear to have been limited to within 10 degrees of latitude north and south of Pangaea's equator. And coal deposits in this region suggest the hot and humid conditions of the great Carboniferous rainforests and huge freshwater swamps. Geological features of Permian rock formations, including decreasing coal deposits and a proliferation of seed-bearing plant fossils, indicate that as temperatures increased, global climate became drier and more seasonal. Ecosystems that once were wet year-round became dry for parts of the year and likely resembled dry savannas. Geographic reconstructions from this era suggest a segregation of equatorial tropical and savanna regions from temperate regions by hot desert that traversed Pangaea both north and south of the equator, effectively isolating pelycosaurs to the tropical equatorial areas that were wet year-round. At this time, the pelycosaurs, such as sphenacodontids like Dimetrodon, were medium to large-sized animals adapted to these ever-wet tropical conditions. Like many living amphibians, they had lower metabolic levels, were ectothermic, and had poor water conservation abilities that prevented their expansion into adjacent drier biomes at higher latitudes. The combination of the adjacent seasonally wet biomes and high atmospheric oxygen levels may have initially facilitated the evolution of higher metabolic rate in carnivorous sphenacodonts, kick-starting the correlated progression towards internal regulation of body temperature and water resources to withstand variable or seasonal climates. These summer wet savanna-adapted therapsids would also have been suited to cooler temperate biomes, but could not expand there due to the mid-latitude hot desert belts. By the mid-Permian, however, the ever-wet tropical forests had likely disappeared, causing the extinction of many pelycosaur lineages. The seasonally dry biomes extended along Pangaea's coast to connect with higher latitude cool temperate biomes. This likely triggered the radiation of therapsids, which had, in the tropical dry savannas, already achieved a physiological condition decently suited to a temperate climate. The likelihood of therapsid radiation originating from a common equatorial starting point is reflected by the fact that very similar taxonomic assemblages of therapsid fossils are found from mid-Permian deposits in both Russia and in South Africa. Some pelycosaur clades also managed to migrate to temperate biomes, such as the herbivorous casiids, but they were at the limits of their range and physiological ability, and so were more prone to extinction. The vacated niches were then occupied by diversifying therapsids. By now we are approaching the end of the Permian period, and our ancestors have made a decent start on the road to endothermic regulation. This road, driven by changing climate and geography, was very complex and involved a correlated progressive evolution of many different characters. Remember, however, that we're not alone on our evolutionary journeys. We share the road with many other lineages. and sometimes they can affect our pathway in very big ways.